Having some trouble. Okay, here we go.
Mute yourself.
Buddy, I'm so excited to be here and to introduce Jordan von Eggers. It has been an enormous honor and privilege to work with this person. If you know Jordy, you know exactly what I mean. And if you don't know Jordy, you're going to know by the end what I mean. Jordy came to us from the University of Washington. I guess that's not quite true because we're about 5-year gap, but she got her a degree in ecology, evolution, and conservation biology. And in the 5 years between when she graduated from the University of Washington and she came here, she did so many amazing things, including working with salmon, working in Alaska, working on colonial water birds, working with sage grouse trapping, and collecting elk poop, which is one of my favorites. Not because I love elk poop, but because Matt Kaufman is here. No, but mainly because it's that she was looking at the DNA in the elk poop, which will become a big theme in a minute. So Jordy has an impressive CV already. She published 5 papers while she was here, and only one of them is part of her dissertation. So she's got 2 more coming from her dissertation. And a lot of her work is that she got involved with, she got involved with what's called the Sedimentary Ancient DNA Society. And 4 of those papers were collaborations with people from that society. And she was an active member of that society and part of the leadership and organization. And she also published a database, which she is first author on, which will be on one of the slides, which is really impressive. So the other thing I was going to say, and Brian will go into this more, but she got to spend 2 months in Uppsala, Sweden, because she needed to learn a bunch of stuff, which we'll get to in a second. And one of her papers resulted from that too. So Jordy's just a go-getter. So I have a couple slides that I wanted to show you. Do I just advance it like this? I'm putting the forward button. Oh, there we go. Takes a village. Okay. So for this work, Jordy works in alpine lakes, and some of the alpine lakes, as you know, in the snowies are accessible by car, but in the winds, there is no such thing because there are no roads. And so all of our trips require backpacking with pack animals, and these were 8-day trips. So I was really lucky when we hired Jordy in 2019, I got to go on a backpacking trip with her, and here she is looking adorable at Cutthroat Lake and sitting with some of my labbies on the bottom. And on the right, you can see she has this big old buff on her face because all of our lakes are at 10,000 feet or above. And so we're exposed to a ton of UV and we get so burned that actually this last year, did I tell you I started getting UV gloves? I'm super into them. Anyway, because your hands get super burnt. So there's Jory with her buff on, and it's not because she's cold, it's because we're so disgusted by getting our lips completely burnt. But all of us wear the buffs or some sort of sun protection, but Jordy took it to a new level. And started, you might not be able to tell because she just looks like she's about to break into a bank, but she's actually wearing her bra on her face. In addition to, underneath it, her buff is still on there. So she's just going big. And on the picture on the right with Annika Walters, where we're dissecting fish, you can see that, where's even her face? The bra is like, we're like, hey, and she's like, this is the best use of a bra I ever had. Anyway, with that, that's my funny anecdote. I'm going to let Brian do more introducing. You're not off the hook.
All right. Well, first off, I just want to thank all of you from zoology and physiology for coming to the geology and geophysics brown bag today. No, but it's been a really fantastic opportunity to work with Jordan because it's given me a chance to think in a very interesting interdisciplinary way. And what Amy was just alluding to is that to do this project, Jordan really had to go outside of all of our comfort zones. She really needed to do, as she has said, she suffered in many ways. Some of that you can see up here. There was a lot of really incredible remote field work. She did work in the Snowies and in the Winds, in the Tetons, in the Sierras, in the Cascades. Most of those lakes didn't have trails to them, at least many of them didn't. It involved carrying all of her equipment, the boats, the coring devices that you'll see, up over steep talus slopes and up at least one place. It felt like third class, at least climbing to me to get up to this one lake. And so Jordy was a champion at getting out to all these incredibly hard field sites. But that wasn't the real extent of her suffering. She also spent a huge amount of time troubleshooting both lab analyses and the statistical work that she ended up doing. And I was so glad and proud of her that she was able to go to Sweden, spend some extended time there to really learn some of these sedimentary DNA methods. And I'll just point out, sedimentary DNA is not a traditional method in paleoecology. So by looking at the past, it requires us usually to sift out little organisms, their remains from the sediments. But the DNA creates this whole new door to like all kinds of new things we've not known about. We have not been able to reconstruct from the past. And so Jordan really explored the methods to do that in a tremendous way. And there was a lot of trial and error. But she got things set up here in the lab that worked really well. And she then also spent a lot of time figuring out all the supercomputing you need to manage all the DNA work. As well as, you're not even going to hear about this, she did this tremendous amount of interesting null modeling to test some of the community ecology ideas. And that was also a giant computational lift. So I just want to say, Jordan did this incredible job of being ambitious in the field, ambitious in the lab, and ambitious with her statistical analyses to go after the questions that she's done. And I'm so proud of her. It's been such a pleasure to get to work with her on all of this. I don't have the funny anecdotes but I will just say I've been massively jealous at all the places that Jordan got to go. So with that, I'll turn it over to Jordan von Eggers, who I'm very excited to have here today.
One day I don't wear a belt, but thankfully Sarah had something. Okay.
Normally, PhDs in paleoecology might have, like, say, 6 links involved. You're going to see data from about 50 different cohorts. So just to give you a sense of the spectrum.
Well, actually, the target almost 7 o'clock. Put the address on the board while Jordan started. Just talk.
Amy's giving the address for the party tonight and has invited everyone in the room and everyone online. Just kidding. Okay. Okay. Oop. See how this goes. Making it work. Okay, can everyone hear me okay? Yeah. Okay, and then let me try this side. Okay, I think that's good. Lots of likes on Zoom. Whew, okay. So as you just heard, my name is Jordan von Eggers. I'm a PhD candidate in the program in ecology and evolution and also in geology and geophysics. My 2 advisors are Amy Bryan, which you just heard from. And today I'm going to be giving my dissertation defense talk titled The Biodiversity of Lower Trophic Levels in Alpine Lakes During the Anthropocene. No, let me try this one. Okay. So I just want to take a moment to remember Dr. Sarah Collins, who we lost last month. She was a really warm and wonderful person. I really admired her as a friend and a mentor. And she was a freshwater enthusiast. And so I'm dedicating this talk today to her. Okay, let's hope I can get this clicker working. If not, okay.
Is there a way to go?
Yeah, I'll just do that, I think. I'm like, perfect. Okay. So my dissertation focuses on 2 overarching questions. First, what shapes biogeography across space and time? And second, how are human activities affecting these biodiversity patterns? So to start, what do I mean by biogeography? So biogeography focuses on the distribution of biodiversity over time and space. And a classic example we think of is the world's biomes here, which are shaped by broad climatic conditions, vegetation, and the species that inhabit them. But this is large organisms, so macroorganisms, or plants and animals. But what about microorganisms? Do they behave in the same way? So we know that the biodiversity of microorganisms is really vast. And just to show you this, this is free living protists drawn to scale on a pinhead. And we see that there are lots of different shapes and sizes. And the biogeography of smaller organisms really hasn't been as studied as extensively in favor of those larger plants and animals. But they play really important roles in all ecosystems and all the biomes we just saw. And then for our second question, we want to know how humans are impacting biodiversity. And so we know globally we're having declines of biodiversity, and this slide shows these different scenarios and future trajectories. But what is really important is this historical long-term background, because this lets us know, have we changed these ecosystems? And if so, have we changed them outside of the realm of natural variability? And so this is where lakes come in. So Brian did get to go to a few cool lakes. He's right down here. Alpine lakes are these really great ecosystems to study biogeography and the controls on biodiversity because they're globally distributed. They're really well contained and well mixed. They create these networks of lakes with varying degrees of dispersal or flow of organisms between them. And lastly, high elevation ecosystems like alpine lakes generally have really few species, and so they're really vulnerable and sensitive to change. I forgot how dry it is here. So lakes accumulate sediments on their bottom, at the lake bottom, which act as these natural recorders or memory banks of past ecosystems. And lakes contain multiple trophic levels, but these lower trophic levels over here on the right are really important because they fill these ecological roles, including primary production, degradation and storage of carbon, and biogeochemical cycling of nutrients. And I'll talk a little bit more about the critters in just a moment. But these organisms on the lower trophic level side are short-lived, and they have really dynamic community structures, and so they respond rapidly to environmental change. And they're affected by things like the climate, lake and water characteristics, dispersal, and also anthropogenic stressors. And so I focus on how the biodiversity of these seemingly pristine lakes are affected by anthropogenic stressors. And these stressors are warming from climate change, nitrogen deposition, and invasive species. In our case, we're talking about introduced trout that have been introduced into these lakes by hand or on foot, and by horses, and also with helicopters more recently. And so here are some of the organisms from lower trophic levels in lakes. Down here on the bottom, we have archaea and bacteria. And so these are microscopic organisms that we're going to refer to as microbes or microorganisms. And when we think about the three domains of life, oops, let me just click this off really quick. This comprises two of the three domains of life in archaea and bacteria. And these top three groups, they are all within eukarya, and they're made up of phytoplankton, heterotrophic protists and rotifers, which are small consumers, and zooplankton. And I'm going to refer to these top three as plankton for simplicity today. Okay, sorry. And so the lake sediment archives signals of past ecosystem by storing the remains of organisms in the sediment that, as Brian talked about, we can go in and visually pick out, identify, and count. And when we think about our trophic triangle over here, for example, some things like diatoms preserve really well, and they've been used a lot in sediment work, whereas things like organisms like copepods really have been absent from paleo reconstructions. And so this really creates this bias towards taxa that preserve well and leave out taxa that could have a really important ecological role, but they don't preserve well, or they're too small or vast to be counted by hand, like microbes. But the DNA of organisms from within and surrounding the lake is stored in the sediment for centuries and millennia. And so recent advances in molecular techniques now allow us to go in and extract the DNA and really unlocked the full diversity of organisms in the lake, including those lower trophic levels we talked about. And so this new really cool tool allows us to ask new questions and glean new insights that we really didn't have access to before. And this is a really rapidly developing field. And so while I'm asking these broad ecological questions, my work also focuses on answering some methodological questions, such as, can we reconstruct zooplankton community from the past few hundred years? And spoiler alert, we can, thank God. Okay, so to demonstrate where this field is at, this is an inventory I put together with some folks in the Sedimentary DNA Society. And you can see that it is quite sparse, although this is a later version, so there's more sites. But it's sparse and there's lots of room to grow, especially when you compare it to this other paleoecology database, Neotoma, that uses more traditional methods, so those physical remains and some more established techniques. And so there's really a lot of momentum to use sedimentary DNA to reveal this rich history of biodiversity across these recent and also geological timescales. And so while I'm addressing new questions about biogeography and biodiversity in the Western United States, my work will also contribute to this growing network of sequences and sites that we can use to examine ecosystem changes on a really global scale. Okay. So here are the three main questions for my dissertation. First, we want to know how microbial biogeography is organized both vertically with sediment depth and then also across multiple mountain ranges, and we're looking at that in Wyoming. Next, I track the biodiversity of zooplankton in alpine lakes, and I'm going to be looking at sets of adjacent lakes in Wyoming. And then lastly, we expand out to look at how anthropogenic stressors are affecting the whole, most of the zooplankton community across the Western United States over the past few hundred years. Okay, so let's get into chapter 1. So microbes and sediments function as these decomposers and transformers of nutrients like carbon, nitrogen, and sulfur, and also other elements like magnesium, and iron. And microbes in sediments have been shown to reflect both current and past conditions. But for the focus of this talk today, we're just going to be focusing on the living organisms, the living microbes in the sediments, and not what we can learn about the past from them. Yet, as we talked about, we know so little about how they're assembled across these really large spatial extents, and particularly with sediment depth below ground. And so microbes make up about 14% of the total biomass on Earth, and 80% of this is stored in these subsurface communities. However, vertical depth is really often overlooked in favor of communities on the surface, but they play these really critical roles, and we don't really know as much about what governs them, where processes could be different because we have burial. So we want to understand the controls of microbes on these two spatial dimensions. And to illustrate this, we're looking at two sediment cores with very different colors, if you can see. And we're also looking at, in this example, cores from two different lakes in different mountain ranges. And so we're going to set up the hypotheses for this. So first, along this vertical dimension, we could have similars within the core, or we could have communities within the core that are really similar all throughout. And so that would suggest weak selection. Or we could have unique communities that are really different between the top and the bottom, and that would suggest strong selection. And then when we look across our geographic dimension, we could have communities that are really similar. So both of these cores could be microbes in the cores could be really similar, and that would suggest high dispersal. And these microbes can move on the wind, on other organisms, and through the water. Or we could have these two cores could be really distinct from one another. And so that would suggest low dispersal and that the microbes can't make it to all these different lakes. And if none of that seems to be the case, it could be random chance or stochastic processes that are best describing what we see in the sediments. So I had a set of wonderful, lovely collaborators, Nathan, John, Eric, Dulcinea, Amy, and Brian. And we published this in Environmental Microbiology in 2024. And so we sampled across the state of Wyoming. Here we're showing the 4 mountain ranges where we took cores, and we collected 48 cores from 36 lakes. And these points represent where we took cores, and the darker blue points represent deeper lakes. And so we did carbon and nitrogen analysis along with 16S metabarcoding of the V4 region. And so here's what we found. So on the right, I'm showing the top 5 dominant families or phyla within our data set. And how we can look at this is depth on the y-axis, which matches our core over here. And then, y-axis, sorry, and x-axis is relative abundance of each of these groups. The gray lines are, they represent individual cores, and the colored line represents the average abundance across cores. And so based on these microbial communities, we found evidence for 3 really distinct zones in the sediment: the redox, transition, and depauperate zones. And these are marked by this really extreme shift in ecological setting from this really nutrient and oxygen-rich environment at the top to this really low energy anoxic zone down at the bottom. And we found that different organisms were dominant in each of these zones. So for example, undifferentiated proteobacteria and teal is really abundant in the redox zones, but then declines in abundance as you get deeper in the sediment. Whereas these brown lines represent taxa that are at low abundance in the surface or the redox zone, and then become more abundant with depth. And I'll note that this triangle represents archaea, so this is that entirely separate domain from bacteria. And so while I'm showing the top 5 groups here, we found these characteristic patterns with depth across the entire data set, all the way from phyla down to individual species. And what you'll note is that it's a little messy, but there isn't difference between mountain ranges. And so what our big bumper sticker for this result is that we found really big differences vertically in the sediment core, but not very big differences geographically. So when we go back to our hypotheses, first, we can rule out random chance because we saw really strong patterns. Next, we saw that these lakes were really similar across all the mountain ranges, so we can rule out low dispersal. And lastly, we saw that there was really different, I probably should let you see what it says, really different communities between the top and the bottom, which rules out this weak selection. So we end up with this high dispersal and strong selection. And I'm kind of going through it simply here, but we, as Brian said, did some really computationally intensive compositional and phylogenetic null modeling to support this. But for sake of time and for everyone's sanity, we left that out today. And so while these results may seem obvious as we walked through them, these are 6 sediment cores, and they show just how different these sediments are. And so we really didn't know what we would find. And I think it's shocking that there's, within all these different sediments, there's the same microbe communities over and over. Okay, so we found that microbes and sediments broke out into these 3 different groups, and were really under a lot of selection. And so these are really distinct zones. So up in the top here, it's like a buffet style eating, and at the bottom would be like the potato famine. Love that one. So when we think about microbes across these mountain ranges, we see that they're really widespread, and we see the same pattern regardless of where they are. And over the years, I've had the hardest time making this chapter accessible to everyone. And so I want to leave you with this idea that microbes have wings. So some of the organisms that we found at the bottom of our sediment cores are also found at the bottom of the ocean floor. And so they really have no problem getting anywhere. And so you can think that we have lake sediment microbial communities floating in the air all around us. We just need the right environment. Okay, so we just learned a little bit about what shapes the biogeography of microorganisms. Now we're going to switch to talking about how anthropogenic stressors affect zooplankton in lakes. And we're looking at living microbes in the sediment, but for the rest of the talk, we're just going to be focusing on organisms that are dead, and we're looking at their DNA that reflects the past. So as I talked about a little bit earlier, these mountain lakes are affected by anthropogenic stressors. But in this chapter, we're just going to talk about the impacts of fish introduction. Okay, so alpine lakes are cold. They're fishless naturally, and they possess few nutrients. And honestly, it's really hard to imagine anything living in this lake, especially when it's only like ice free for like a few months of the year. I think this is like mid or early July. And but surprisingly, there are a ton of critters in this lake. And we know that this is Casey in this picture because she's the only one epic enough to hike all day, every day in chacos and on terrain like this. So I'm wearing chacos in this video here, but I put them on after I got to the lake like a normal person. And so here I'm going to show you the dominant zooplankton in this lake. Okay, so you can see that it is a really efficient swimmer. And it's a really effective phytoplankton consumer, so it contributes to why these lakes are clear and oligotrophic. And it's also a predator of other zooplankton. And so it's really the top dog in these lakes. And remember how we talked about some organisms preserve better than others? Copepods do not preserve well, so they've really been left out of community reconstructions. And so across the Western United States, 95% of all mountain lakes were naturally fishless. But starting in the 1900s, we've been introducing them with trout. And now 60% of all mountain lakes and 95% of the deeper, larger lakes contain trout. And so when you introduce trout, they eat all the large-bodied zooplankton. And how they do this is they have these gill rakers that act as sieves. And they catch all the large zooplankton, and then the small zooplankton are able to filter through uneaten. And this isn't a trout, but this just represents this really well. And so after fish introductions, we see the switch to small-bodied zooplankton. And these small zooplankton are not as good at eating the zooplankton, so we tend to see higher abundances of algae or phytoplankton. And so this manifests as a top-down effect or a trophic cascade. And so this chapter has two goals. First, to determine baseline conditions prior to fish introductions. And then second, we want to measure the timing, magnitude, and direction and rate of change in response to fish introductions. So I had a wonderful set of collaborators for this. We've got Brian, Lindsay, Katie, Annika, and Amy, and we're in between submissions right now. And so here are all the lakes we cored in the two mountain ranges. And so the green lakes here are fishless lakes, and the purple lakes are lakes with fish introductions, but note that they were stocked at different times all the way from 1928 to 1974. And this was supposed to be our fishless lake for comparison, but this is the perfect application of this. We looked at the DNA and we're like, something doesn't look right. And so we actually went back and got more records and found out that indeed this lake had been stocked. So this is another fish lake. And these lakes were stocked with brook trout and cutthroat trout. And so with the help of my many collaborators and graduate students, we hiked out with our llamas and we hopped into these boats, and I collected sediment cores and we extruded them on the shore like a push pop, and then put them into these little bags. And then the DNA we flash froze on dry ice. Okay, and then the methods, I contrasted fish and fishless lakes and dated the sediments using lead-210 and looked at zooplankton metabarcoding using 18S V9. And we did a lot of other analyses with these cores, but for this talk, I'm just gonna focus on the main methods and findings. Okay, so we talked a little bit about the size differences between zooplankton, but there's also these two main groups. And unfortunately, I don't have a lot of time to go over their really cool biology, but just note that there's these two groups we have and that they come in a range of sizes. So here's all the copepods, And these are the cladocerans. And this is that big red copepod we saw in the video earlier. Okay, so now we have all the pieces to the puzzle. We're going to look at what we found. So along the x-axis, we have time going from left to right. On the y-axis, we have relative abundance. And we're looking at these 4 groups: large-bodied copepods, large-bodied cladocerans, small-bodied copepods, and small-bodied cladocerans. And we're looking at our 3 fishless lakes. So what we see right away is throughout the entire record, these big red copepods are dominating. And you can see that with the red line that's near 100%. But when you introduce fish, which is shown by this vertical line here, we see a really rapid shift to these small-bodied cladocerans, and then also a shift to small-bodied copepods in more recent decades. So here, I have to put it into pictures. So these copepods, the large-bodied copepods, play these really key ecological roles as a keystone species. And these smaller zooplankton really just can't fill that same role. And we think that Daphnia in particular increase after fish introductions, these small-bodied Daphnia, because fish can increase the amount of phosphorus available in the lakes, and Daphnia are able to outcompete other zooplankton in high phosphorus environments. And just what's really remarkable about this is that without sedimentary DNA, we wouldn't know what was going on in the lake because we would totally miss what's going on with this big red copepod. And so what we found is that fish introductions ended centuries of long-term stability dominating by these large red copepods, which I'll mention they're Hesperodioptimus. And we saw the shift to small-bodied cladocerans. And if you remember one thing today, I hope you remember something inspired by my dad, was that these fish are cuckoo for cocopods. Oh, I love it. Okay. So we just learned about the impacts of fish introduction on zooplankton. Now we're going to zoom out and we're going to talk about multiple anthropogenic stressors across multiple trophic levels in multiple regions across multiple centuries. Okay, so in addition to those really dramatic effects of fish introductions, we also have warming and nitrogen affecting these lakes, nitrogen deposition. And so let's quickly revisit the cast of characters. We're going to start with zooplankton because we just learned about them. So they eat heterotrophic protists and rotifers and phytoplankton. And we also have these heterotrophic protists and rotifers. So these are smaller consumers and they consume organic matter. So they consume other organisms, they consume phytoplankton and bacteria, and they also eat dead material, and they also can be parasites. And then lastly, we have phytoplankton. And so this includes both autotrophic organisms, so ones that can photosynthesize, but also mixotrophs that can switch between photosynthesizing and heterotrophy, which is eating other organisms. And if I'll remind you that all 3 of these we're calling plankton. And so we talked about fish introduction already. But now we're going to talk about nitrogen deposition. So I'll remind you that these lakes naturally have low nutrients. And so here on the right, we're looking at atmospheric nitrogen deposition across the lower 48. And I've circled our 3 study sites. And so atmospheric nitrogen deposition comes from industrialization and agriculture. And you can see right away, the east is a lot higher than the west. But when you look at the west, it looks like a topographic map, but it's actually not. It's just because this nitrogen accumulates in these mountain lakes because the winter months where a lot of this accumulates and falls as precipitation as snow and gets, builds up in these systems. And especially these high elevation lakes are naturally in settings with low nutrients, and so we're really just pumping them full of nitrogen. And nutrients have consequences for overall primary productivity. So it can change the composition of the phytoplankton communities, but it also can increase their abundance as well. And these can have effects that kind of filter up through the trophic levels. So this is our bottom-up stressor. And our last stressor is climate change. And so we know that temperatures are warming, but this map nicely shows that some places are warming quicker than others. And globally, ice-covered lakes are warming quicker than ice-free lakes. And that's because of changes in that ice-free season, changes in thermal stratification, and also interactions with things like radiation and cloud cover. And climate can have effects on trophic levels in multiple ways. So it can increase the rate of increase the metabolic rate of our primary producers. It can affect the growing and reproductive season of both primary producers and consumers, and it can place physiological stress on any of these taxa. And these effects can kind of propagate throughout the trophic system. Okay, so we have our 3 anthropogenic stressors and their potential effects on mountain lake plankton. This last chapter was with my 2 lovely advisors, Amy and Bryan. And so I sampled across the Western United States in California, Washington, and Wyoming, and I collected 9 cores with that had fish in the lake and 5 without. And it was actually a lot harder to find lakes that were fishless. And here in the parentheses are when the fish were first introduced. And here are some examples of lakes from these regions from pictures that I took, because I just need another excuse to show you more pictures of lakes. They look cool? Cool. And so this time we didn't go out with pack animals. We went out with a small army of graduate students, family, and friends. And I'll just say a few things about some of these trips. So this trip, we were in Grand Teton National Park, and we were soaked about 15 minutes in, and I had to sleep with a wet sleeping bag. This one, my cousin Lee didn't have a rain jacket, so we used a trash bag. Worked okay. This trip, I puked going over Kearsarge Path both ways, so once going over and then once going back the next day. This trip, Lorelai and I really didn't want to bring a second tent. And so Sue said she would cowboy camp and sleep outside. But then the second night we had a crazy bad storm. And so the 3 of us were in a 2-person tent that really only fits like 1 person comfortably. And this last trip in Mount Rainier with Ian and Danielle and Eric. Eric's my husband. Danielle was recovering from a broken collarbone with our incredibly heavy packs. And this is the trip that Eric looks happy here, but this is when he instated his 5-mile limit per day. This is after we just did 24 miles in 24 hours. So a huge thank you to all these people that helped me core. And so the methods are pretty similar to chapter 2, but we added a few things. So we're looking at fish and fishless lakes across these 3 regions. We dated the sediments using lead 210. We're using DNA metabarcoding to look at plankton. And we also added air temperatures from prison to look at warming. And then I also measured nitrogen isotopes to track atmospheric nitrogen deposition. Okay, so here are some of our results. So again, on the x-axis, we have time going from left to right. And here we're looking at temperature across all of our lakes in these 3 regions. So we're looking at temperature, and it's we're looking at it as an anomaly or a difference from a mean. And so what we see is that we have increases in temperature in all 3 of these regions starting in the late 1900s. And note that Washington has a smaller increase than in Wyoming and California. And now we're looking at our nitrogen deposition data. So again, we're looking at this as an anomaly from mean. And what we see is that we start seeing increases in atmospheric nitrogen deposition starting around 1900. And again, Washington has less deposition than places like California. Okay, so now we're going to look at all the stressors alongside our plankton data. So this is what I just showed you in the slide before, but now we're splitting it by fishless lakes and fish lakes. And so we see in both sets of lakes, we've got warming temperatures and increased nitrogen deposition. And then on this right panel, we have these vertical lines that represent when fish were introduced. And since we're looking at 9 lakes, we have 9 lines. And so I identified the dominant groups or taxa and ran linear mixed effects models using the stressors as fixed effects and lake as random effects. We also incorporated autocorrelation structure in the residuals. And I'm going to start by going through the top of that trophic triangle. So here we're looking at zooplankton. And we have our large-bodied zooplankton and our small-bodied zooplankton. And so what we see in the fishless lakes is that the large-bodied zooplankton are dominating throughout the record, and small-bodied zooplankton have really low abundances. But just like we saw in chapter 2, after fish introductions, we see a decline in these large-bodied zooplankton and an increase in the small-bodied zooplankton after fish were introduced. And so here, fish are the driving stressor for these communities, or for these groups, sorry. And next, when we look at heterotrophic protists, when we're looking at ciliates, it's a little messy here, but we see that they both kind of increase over time, suggesting that these regional stressors may be more affecting them more than things like fish introduction. And so our models indicate that nitrogen is related to the changes we see in ciliates. And now we're looking at phytoplankton, and we're looking at dinoflagellates and gyrista. And this includes things like, gyrista includes diatoms and eustigmatophytes. And so we see in both sets of fishless and fish lakes, we see increases in both of these groups, which are related to nitrogen and warming. And lastly, we're looking at streptophytes and chlorophytes here. We see that they both decline regardless of whether they're in fish or fishless lakes, and suggesting these regional stressors are driving them both. And we see that they're related to nitrogen and warming in nitrogen. So when we take it all in, we see that fish are affecting these large-bodied zooplankton, affecting the zooplankton, nitrogen is affecting the protists, and nitrogen warming is affecting the phytoplankton. And so when we think back to our hypotheses, we see that fish have the strongest effect on zooplankton, strongest top-down effects. We see that nitrogen has strong effects on phytoplankton and the protists, and multi-level effects from warming are most strongly affecting phytoplankton. So we just looked at some of the really abundant groups in over time and how they're related to these stressors. But now we're going to zoom out and look at the entire plankton community and how they're related to the stressors. Okay, so the best way to show this is an NMDS plot. And so it essentially takes the entire composition of the community and distills it down into one point. And it places it on this plot depending on how similar it is to the other communities. So 2 points close together are going to indicate really similar communities. So these would be 2 different communities in time from the same lake. And if they're farther apart, this indicates that they're more dissimilar from one another. Okay, so here are all of our lakes. And whether or not there's a black outline around the point indicates whether or not there was fish present in the lake with that community. And so we're gonna look at zooplankton, and I'm just gonna put up one lake to start. Okay, so here we just have footprint lake. And the size of the point relates to time, with larger points being older and smaller points being closer more to modern. And so this ends up kind of splitting this as past and modern. But really what we see right away is that these past communities all cluster together and they don't have fish. And after fish introduced, it pushes these communities into this new zone in more recent decades. Okay, so now I'm gonna put up the rest of the lakes. This is Wyoming, Washington, and California. And so for zooplankton, what we see is that a lot of these communities in the past were dominated by that big red copepod. But over time, and as we added fish, we see a shift to these small-bodied zooplankton, but which taxa depends on the region. So we see that fish are working to diversify these zooplankton communities, or differentiate them and make them more different. And we also regress these NMDS scores and did tests against our stressors as well to confirm these relationships. Okay, so now we're looking at heterotrophic protists and rotifers. And what you see here, it's a little bit different. So we see some geographic separation with Wyoming and California kind of clustering together in the past, and then Wyoming clustering down here in the bottom. And what we see is that all these communities, or all these lakes, all these communities from these lakes homogenize over time and become more similar. And this is regardless of whether or not fish are in the lake. So this suggests that these regional stressors are really driving the changes we see in the protists and rotifers. And lastly, we're going to look at phytoplankton. And here, the geographic separation is really clear. So we've got Washington up at the top, it's California in the middle, and Wyoming down at the bottom. I note that, again, they homogenize over time, and they all become more similar regardless of whether there's fish there. And note, too, that remember how we talked about Washington had lower increases in warming and nitrogen deposition? Well, we actually saw smaller changes in the community as a whole compared to Wyoming and California. And so this suggests that stressor intensity could also affect how much these communities are changing. So when we put this all together, we see this consistent transformation of plankton communities across the West, but that the outcomes really depend on their trophic level. And so when we think back to our hypotheses in the framework, we see that top-down effects work to differentiate zooplankton communities, while bottom-up and multi-level effects work to homogenize protists, rotifers, and phytoplankton. And so clearly these human activities that we've done have pushed these communities into new zones and outside of their historic realm of natural variability. And Brian had a really great analogy for this. It's like taking a Rubik's Cube and shuffling it up. We have these new ecological outcomes that we really don't know the consequences of yet. Another lake. So to wrap this all up, for our methodological conclusion, we found that sedimentary DNA is this really powerful tool that we can study the biodiversity and biogeography of organisms from microbes all the way up to zooplankton. And for our ecological conclusions, we're looking at the biogeography of these lower trophic levels. And we found that sediment depth really shapes microbial communities consistently across mountain ranges. We found that fish introductions rapidly reorganize zooplankton communities. And we found that these anthropogenic stressors consistently transform these zooplankton communities, but the outcome depended on your trophic level that you're in. So I have a lot of thank yous. I also have a backup plan that if I start crying, I'm making Niall read all my thank yous. So I've worked hard on them. Okay, so I have a lot of thank yous to give, but I'll try to be quick. So I want to thank our funding sources. I want to thank National Park and Forest Service. I want to thank all my lab mates and all the people who helped me collect cores out in the field. I want to thank the Sedimentary DNA Society. They were such a fun community to be able to be a part of. I want to thank my committee, Ellen, Annika, and Katie. These three rock star lady scientists were such a joy to get to interact with, and I loved getting to learn from them. I want to give a shout out to all my Lair amigos. We made so many fun memories here that I'll never forget. I'm really thankful for my family members whose love and support has enriched my life every day. And a special thank you to my parents who instilled the love of learning in me at a really young age and have been encouraging and supporting for my whole 20 years of academic pursuit. I want to thank my husband, Eric. My mom always told me that life can be good or life can be great, and it's worth waiting for the right person. And with Eric, I couldn't imagine life any better. Eric is insanely goofy. He's really social to the point that it's annoying. He's really inquisitive, and he's just an outstanding partner and an amazing father to our sweet little pumpkin. And Eric, I want to thank you for years of support and love in all facets of my life and career. And thank you for moving out to Laramie to end our many years of long distance. Now to the big guns. Okay. I want to thank my two advisors, Amy and Brian. I really can't speak more highly of these two people. So the science is what drew me in here, but the real reason I stayed and switched from a master's to a PhD is because of Amy and Bryan. I really wanted to learn from them. I wanted to create cool science with them, and I really wanted, don't look at me, Amy. Don't, I was doing so good. I really just wanted to be a part of both of their orbits. Okay, look at the screen. Okay, so I want to say a few things about Amy. So her enthusiasm and vigor for life is just truly unmatched, as is her love of small organisms that are often overlooked. She loves these organisms almost as much as she loves her chickens. And don't worry, Amy, I got you a gift that has chickens on it. And it's specifically for you and your chickens. So Amy's really given me a deeper appreciation of freshwater organisms through our many meetings where she would geek out on their cool biology, as she puts it. And as many of you know, Amy's laugh can be heard almost anywhere if you're in the same building with her. And you really just can't help but get wrapped up in her positive and open-minded energy. And Amy really sees the best in people, despite the many hardships that she's endured while I've been here, and she really finds joy in every nook and cranny. Amy's an excellent writer, and she taught me the importance of being direct and concise, although I'm being pretty wordy today. She's brilliant, she's creative, and she's incredibly resourceful. But most of all, she's just a gem of a human I'm really happy I got to get close to. I can do this. And Bryan really cares deeply for people, and he's quick to connect on any level with those around him. He fosters a really inclusive environment that genuinely appreciates and encourages every perspective. And Bryan really brings science to life. He has an amazing memory and recall. And Bryan, with an expo pen and a whiteboard, is truly unstoppable. I can't count how many times he's taken these really, really complex ideas and concepts and distilled them into these really simple and transferable ideas, which is just the perfect definition of science communication. And even without his expo pen, Bryan really brings the natural world to life. So on this trip, one night we sat under the stars and he just talked to us about astrology. And then we would sit on the shore, he would explain the glaciers, how they worked, how you can tell which ways they receded by the scours on the rocks. And so he really helped create this deeper appreciation for ecosystems in me, but also what we can learn from the past. And Bryan is really just truly in his element when he's brainstorming, interpreting data, and collaborating. And so while I've learned so much about science from Bryan, I've really learned what it means to be a consistently kind and thoughtful human. So these two have had such a substantial impact on my life, and I'm really sad that I'm not going to be under their mentorship, but I know that we'll continue on as good friends. And I can't end this talk, I have to apologize, without showing this picture of when Amy dressed up as a planarian. And she didn't realize that it looked like a very specific male body part. I'm sorry. Okay, so with that, I know we don't have a lot of time. Should I leave this up? I'll leave this up. With that, I'll take questions. My face hurts. Okay, fine. I did. I got one more. Yeah. Yeah. It's huge. I can check the chat too. Yes.
In your you talked about like the high selective pressures causing that sort of vertical diversity. What are some of those selective forces?
Yeah, it's really cool. So the question is, what are some of the selective forces on the microbial communities and the sediments? And so it's really remarkable how it works. So in the top of the core, you have a lot of like nutrients and energy. and there's oxygen. But as you go deeper into the core, you actually follow this this chain of electron acceptors. So essentially, it's like what these taxa can use. So it goes from like oxygen, and then they like reduce nitrate, so they do denitrification, and then they're doing sulfur, and then by the end, they really can only do things like methanogenesis, which is in anoxic conditions. And so you see taxa that are doing that down at the bottom. Yes.
In chapter 2, just one of the ocean fishes lakes, you have little cliffs that like go away. Thought about like what those are and why those do lead to like bigger ships.
Yeah, are you talking about like in the past with just the copepods or after fish injection? Yeah. Before and after some of the variations. Yeah, I don't know. We did for that chapter also look at nitrogen deposition and warming too, but I just didn't talk about it. But the blips you're talking about too are kind of before that. I don't really know why. Maybe there's just like one taxa that was able to get really abundant. Yeah, that's a really good question though. Nothing that we found that we knew explains some of that. Yes, Dave?
Fantastic.
Awesome.
You presented the lakes as being driven exclusively by primary productivity from the phytoplankton.
Mm-hmm.
This picture shows a lot of surrounding vegetation. Surrounding vegetation is a flux of organic energy into the lakes, and how does that vary across space and time? Do you have any idea about that?
Yeah, definitely. Like, there's lots of work that shows, like, especially because if there is a lot of this material, maybe it's kind of set and is like continually giving certain inputs into the lake. But especially with things like warming, there's a lot that it's showing like there's more dissolved organic carbon in these lakes, and also like the tree lines moving up. So maybe there's more organic material washing in. Yeah, it would take, I think, more exploration into understanding like how much inputs, and does that differ by lake, and is that what's controlling the phytoplankton? Definitely, that'd be super cool to look at. But yeah. You guys, I studied really hard for all these questions. Probably more than I did this. Yes, what?
Very little different species, or very similar species composition. Could that just be like at the level, the taxon level, or is it like species, would there be a lot more variation?
Yeah, there definitely is. And I didn't talk about this today, but there's sometimes differences between lakes too, depending on like the deeper, cooler lakes tend to have taxa that are a little bit different than the shallow, warm lakes. So yeah, there is some more variability, but we tend to see kind of these big groups that are like kind of doing similar functions in those zones. But yeah, there's more nuance. You didn't want me to go over that today though. Yes?
Third chapter, you talk about diversification and then kind of the lakes across different states becoming more similar.
Mm-hmm.
And is that, did you look into at all what that means? Like, because I feel like each individual lake become more diverse, but if it's diverse in the same way as all the lakes become more similar. So is it that forming separate types of communities? Is it that they're really specialized in one type? Does that make sense?
Yeah. So is it like
diversity within a lake versus diversity across the state?
Yeah. Okay. So like the question is, what's driving the diversity across the lakes versus could there be also diversity within a lake that's being driven by different things?
Yeah, so that lakes could become more similar, but with those lakes could all just be very diverse versus they could all be very different, and each lake has very specific, not diverse things. So they're all different, but yeah, where is the diversity coming? Like what level? I guess.
Yeah.
What the community compositions look like, between those lakes, or what are they all homogenizing towards?
Oh, okay. Sorry. So is it like, why are they changing in a similar way?
Yeah. Like, yeah.
I'm sorry.
Like, what does that actually show you the diversity, I guess?
Yeah. Yeah. Cause right. It kind of seems like the bottom's broken up by time, but then geographically they become more similar. I think it could be, and I need to explore this a little bit more because honestly, I was overwhelmed by all the taxa that were in there. But I think that it switches towards taxa that are able to like capitalize on nutrients. So like, For example, there's one organism, Nanochloropsis, that's like really good at, it's like an algae that dominates in oligotrophic lakes, but then we see that that declines over time. And so then I think it's like, it's becoming more similar because there's these taxa that are able to capitalize on these nutrients, and they're kind of overwhelming maybe this larger diversity they had in the past in these lakes. And then that's kind of my understanding of it. I hope I answered your question. If not, we'll talk after. Oh, thank you. I hope everyone on Zoom can hear the questions. I forgot to repeat them. So.
Let's thank Jordy.
Thank you. Thank you to everyone on Zoom. Thank you.
Oh.
I gotta turn this